Hello, everyone. So welcome to the monthly webinar of Next Generation Vaccinologists. Today we have two speakers, but before we start, I would like to remind you that we have our next webinar 6th of March. So make sure to join that too. And in the link in the chat, you can find a link to our LinkedIn page. So if you would like to be part of our group, help us with organizing webinars, increase the visibility of next generation vaccinologists. You can also be part of this group and get involved. So please let us know. During the talks, you can also put your questions down in the Q&A part. And then at the end of the first presentation, we will read those questions, and then we move on to the second presentation. And at the end of that, we will also answer your questions. So you can do that during the webinar. So with that, I would like to start introducing James Allen. So he did his PhD in the University of Georgia, finished in 2021. And at the moment, he's in Cleveland Clinic as a research associate in the group of Dr. Ted Ross. And today he will tell us about longitudinal assessment of human immune responses to commercial influenza vaccination. So James, please, thanks for joining us. Floor is yours. You can share your screen and we can start.
Okay, can you see my slides?
Yeah.
Okay, great. Thanks for the nice introduction. As was mentioned, my name is Dr. James Allen. I'm a research associate at the Cleveland Clinic. Our location is actually located in Port St. Lucie, Florida. But I'm a member of Dr. Ted Ross's laboratory. Our lab primarily focuses on designing influenza vaccines that are computationally optimized to enhance breadth of response to drifted antigenic strains. But today, I'm going to be talking about some of our work on assessing human immune responses to commercial influenza vaccination. All of this work is part of the Civics Network, which is the Collaborative Influenza Vaccine Innovation Centers funded by NIAID. And this project is a joint venture that takes place at the University of Georgia and at the Cleveland Clinic. Okay. So I'll give a little bit of background about the influenza virus. It belongs to the viral family Orthomyxoviridae. These are envelope viruses that contain 8 segmented negative sense single-strand RNA genes. They encode for about 14 proteins, and they're characterized by their surface glycoproteins, the HA or hemagglutinin and the NA, the neuraminidase. And these viruses are named according to those glycoproteins. So if a virus has a group 1 HA and a group 1 NA, it's classified as an H1N1 and so forth. There's 3 major types of influenza, A, B, and C. Type A infects primarily humans, birds, and pigs, while type B and C only infects humans. These viruses cause acute respiratory diseases in humans that infect about 5% to 15% of the global population every year, resulting in about 300,000 to 650,000 deaths, most of which being seen in the children and elderly. The influenza virus is an antigenically variable virus that can undergo continual antigenic drift. So what that means is that mutations can occur in the immunodominant proteins on the surface of the virus in order to evade host immune pressure. This primarily takes place in the hemagglutinin and the neuraminidase glycoproteins. And because of this, vaccine efficacy can fluctuate from one season to the next, and often annual reformulation is necessary. So as a result, annual vaccination is currently recommended for everyone greater than 6 months of age. Currently in the United States, influenza-like activity is very, very high. There's only about 3 states with minimal flu activity. There's also about 5 or 6 states that have the highest levels of influenza-like activity. This is much higher than previous years seen. So right now, flu is in its peak in the United States. Influenza vaccines do exist. Standard of care vaccines typically consist of inactivated wild type viruses that are the ones that were most prevalent from the previous season. These are typically administered as either trivalent or quadrivalent formulations, typically containing 1H1, 1H3, or 1 or 2 influenza B strains. The strain selections that go into the vaccine are made approximately 6 months prior to distribution because it takes a long time to make enough doses for a large population of people. But because of this time for manufacturing, is going to allow the virus time to antigenically drift, which can also lead to vaccine mismatches and low protective efficacy within a season. The current estimated protective efficacy of this year's influenza vaccine is about 35 to 40%. So in order to understand better the human response to commercial influenza vaccines, our lab has their own clinical trials that we run here at the Cleveland Clinic and at the University of Georgia. We have some eligibility criteria where people between the ages of 18 and 90 years old, and we're also looking for them to weigh at least 110 pounds and not have received the current influenza vaccine in this season. But when people come into the study, they'll receive the flu vaccine and get their blood drawn. And then there will be 3 to 4 follow-up visits after the initial visit where we collect blood that we use to analyze sera and PBMC populations. So as I mentioned, this is a collaborative effort led by Hannah Hanley at the University of Georgia and Michael Carlock and Brianna Gomez at the Cleveland Clinic. The goal is to perform influenza virus vaccinations in people from the age of 11 to 90. We look at a number of different vaccine types, split and activated, primarily flu zone. That's what mainly I'm going to be talking about today. But we also look at responses to recombinant HA like flu block or live attenuated vaccines like flu mist. And what we do is we collect and process, catalog, ship, and store the samples for assessment in order to look at antibody, B and T cells, innate immunological responses, and omic responses for vaccine effectiveness by age, gender, and race. We use this information to then model vaccine responses to identify potentially new correlates of protection and help guide future vaccine design. And we also want to identify human assessment strategies for our own proposed phase 1 human clinical trials. So like I mentioned, we vaccinate people and then segregate them into different age categories. People come into the study and get their blood drawn at day 0 and receive the vaccine. Then they come back 7 days later and get bled again. And then on either day 21 or day 28 to collect more blood. And we use the serum collected from the blood for HAI and ELISA assays, and PBMCs collected for FACS, ELISpot, in vitro memory B cell differentiation. In certain past seasons, we've collected urine to look at things like gene expression or the proteome. And currently, in the last few years, we've been collecting saliva to look at IgA and transcriptome information. So we've been doing this study for a number of years now. When we first moved to the University of Georgia in 2016, we started what we call the UGA cohort. The ones I'm going to focus in on today is the UGA 1 through the UGA 6 cohort in individuals that were vaccinated with Fluzone. But as you can see at the top of this figure, the vaccine strains change from year to year. Over this 6-year span, the H1N1 vaccine component changed 5 times. The H3 component also changed 5 times, and the influenza B component changed 3 times. This is all work that was published last year in PLOS One by Michael Carlock and myself in Dr. Ross's laboratory. When we look at the patient demographics across UGA 1 through 6, there were a total of 1,800 participants. They were predominantly female, consisting of about 60% and around 40% male. The cohort was also about 70% Caucasian, 10% African American, 5% Hispanic Latino, and 5% Asian. And across these 6 seasons, we also had a cohort of individuals, 243 people that participated in 3 consecutive seasons, so UGA 4 through 6. And the demographics of this cohort lined up pretty well with the overall demographics, again, being about 60% female, 40% male, 90% Caucasian, and then the other races represented within the remaining 10%. The first thing that we do in our primary readout for this is the hemagglutination inhibition assay. This is an assay that was well established and has been around for a long time. It's used to titrate antibody responses to influenza viral infection or vaccination. And it takes advantage of a natural phenomenon that happens when you mix red blood cells with influenza viruses. The viruses will actually bind to the sialic acids on the surface of the red blood cell, causing the red blood cells to sort of clump up and make a lattice-like formation. However, if you add sera that contains antibodies that are specific to binding HA, they can prevent this interaction, and the blood doesn't clump up. So when you tilt the plate, you'll see some wells will run. That means they have enough antibodies to prevent the agglutination, and you'll see other wells that clump up and still have this lattice in there. This is a well-established assay that has correlates of protections associated with it, and I think that's why it's still a gold standard. So if you have an antibody titer of 1 to 40, that correlates with a 50% chance of being protected from that viral infection with that particular strain. What we do is we analyze the sera that we collect from each individual at day 0 and then at day 28 following vaccination. And we perform HAI assays against panels of H1, H3, and influenza B viruses. And then we stratify this data based on the age of the individual. So the way you sort of read these graphs is the more blue the certain cell is, the higher the HAI titer is at that particular time point. And then along the left-hand side of the y-axis, we have the ages of the individuals starting from youngest and going down to the oldest. And you'll sort of see a trend here that the top of these graphs are typically more blue than the bottom. In each column at the top, we have the virus that we tested the HAI assay against, and in red is the vaccine strain from that given season. One of the main things that popped out was that the 11 to 17-year-old age group had the highest HAI titers at day 0. And following vaccination, this age group also had seroprotective titers across the H1N1 panel in each season. Also, if you looked at individuals from one season to the next, they were able to maintain their seroprotective titers, or titers greater than or equal to 1 to 40, well into day 0 of the following season. However, as participants aged, the HAI titers at day 0 began to decrease, and fewer individuals had seroprotective HAI titers against the vaccine strain in the following season. We also saw less back boosting. So can a vaccine from a current season help you elicit antibodies or a memory recall response to previous strains? Those were more pronounced in the younger individuals, whereas the older individuals had trouble mounting immune responses to previous vaccine strains. When we looked at the H3 and 2 responses, we again saw that the youngest individuals had the highest HAI titers at the day 0 sampling time point, and also had seroprotective titers across the H3 panel. The lowest HAI titers were observed against the Kansas 17 strain. Vaccination with this strain induced seroprotective titers following vaccination in the year that it was administered as a vaccine antigen. But those protective titers against Kansas 17 were not well maintained into the next season. And looking at UGA 5 and UGA 6, when individuals were vaccinated with Hong Kong 19 or Tasmania 20, these vaccine antigens didn't really boost the response to Kansas 17 moving backwards. But similar to H1N1, we saw that as participants aged over 35, they had lower HAI titers in each season, both prior to vaccination and post-vaccination compared to younger individuals. For influenza B, unlike for H1 and H3, the B Yamagana component of the vaccine remained the same. It was Phuket 13 across all of the cohorts, while the B Victoria component changed from Colorado 17 to Washington 2019 in the UGA5 cohort. Again, the youngest individuals aged 11 to 17 had the highest HAI titers coming into each season and following vaccination. And as the participants aged, we saw those day zero titers decrease. Fewer individuals had seroprotective HAI titers against the vaccine strain. And this was a little surprising because these older individuals, by the time they were in UGA 6, they had now seen the Phuket 13 antigen in the vaccine at least 3 times. But nevertheless, they still had low day zero HAI titers and struggled to mount seroprotective responses to this vaccine strain. We then analyzed the HAI results looking for a criteria that we call seroconversion. So this is a 4-fold rise in antibody titer. This is usually a pretty strong indicator of whether or not you mounted a good response to the vaccine. And across all 6 seasons, about 30% of individuals had a 4-fold rise in antibody titer to each individual component of the vaccine. One thing that we noticed when we started looking from one season to the next was that in seasons where clade changes were made in the vaccine strain from one season to the next tended to result in a higher seroconversion rate for the vaccinees. We also looked at the amount of people who came into the study as seronegative or had a titer of less than 1 to 40, and then following vaccination became seropositive or had a titer greater than or equal to 1 to 40 to the vaccine strain after immunization. And what we saw was that across all 6 seasons, about 40 to 95% of the seronegative individuals at day 0 mounted a protected immune response following vaccination, at least against the vaccine strain from each year. Certain years were higher. So for H1N1, the Michigan 15 had the highest amount of these seronegative individuals who became seropositive, whereas UGA 5 had the lowest. We then looked at the people who came into each study at day 0 with a seropositive titer, or one that's greater than or equal to 1 to 40. And either stayed seropositive, had a 4-fold rise, or became seronegative following vaccination. And what we saw was that about 75 to 80% of these individuals that came in with a seroprotective titer maintained those seroprotective titers following vaccination, but only about 15 to 20% of those had a 4-fold rise in antibody titer. We speculate that this is because they already came in with a high titer, so there's not a lot of antigenic space for that titer to grow following vaccination. But when we looked at people who were seropositive on day 0 and then became seronegative or had a non-protective titer following vaccination, this only occurred in about 2% of individuals. We then compared the effects of prior vaccination between people who had been vaccinated in 2 consecutive seasons compared to those who had not been vaccinated in the previous 2 years. And the main thing that we found was that individuals who were not vaccinated in the last 2 years had higher rates of being seronegative or coming into the study with not protective titers. But following vaccination, they had a higher rate of seroconversion, that 4-fold rise that I was talking about in antibody titer. Whereas individuals who were vaccinated annually were more likely to enter the study with already having seroprotective titers at day 0 and maintaining those into day 28 following vaccination. Conversely, they were also less likely to have a 4-fold rise in antibody titer following vaccination. So in conclusion, in this study, most of the participants vaccinated with split inactivated influenza vaccine had at least a minimum twofold increase in anti-HA antibody titers within 4 weeks post-vaccination. And age played the largest role out of any other criteria that we looked at in vaccine effectiveness. Most of the older children and young adults had seroprotective HAI titers at day 0 that were either maintained or increased over time following vaccination. And as participants aged, their vaccine-induced antibody titers began to wane over the subsequent year, especially those aged 65 and older. Aging is associated with diminished humoral and cellular immune responses that impact B cell activation and the induction of antigenic T cells. There are high-dose vaccines available to people 65 and older, and about 71% of these participants in our study received the high-dose formulation. So even though they're getting the high-dose formulation, we're still seeing differences in their responses compared to the younger individuals. We also saw that seasons where a clade change was made between vaccine strains led to more people being seronegative at day 0 and more seroconversion events at day 28. Likely because this is an antigenically different strain that their body has not encountered before, but they were able to mount effective antibody responses following vaccination. And then we also saw that participants who had not been vaccinated in the prior 2 years were less likely to enter the study with protective titers. However, their antibody titers rose to protective levels following vaccination, and that people vaccinated in 2+ consecutive seasons were more likely to enter the study with protective titers, but again, were less likely to have a 4-fold rise in titer post-vaccination. So it's important to get vaccinated annually in order to keep your antibody titers at protective levels year-round. And future studies will focus on the differences in immunity that were induced by the different types of commercial vaccines, and also looking at different cellular B and T cell responses elicited by the vaccines. It took a team of people to produce this work, so I'd like to thank all of our members at the University of Georgia, as well as the people at Cleveland Clinic for all their hard work in collecting these samples and performing all these assays. And with that, I will take any questions.
Thank you, James, for this great presentation. It's I'm looking at the panel now. There's no open questions at the moment, but maybe I can ask you a question first. So it was striking when you showed that people above 35 years old, then you already see a big decrease, right? So mostly when we talk about aging, everyone is thinking about much later age groups, but then this is already happening pretty early. But then only the guidelines are including 65+. So maybe there's something to think about there, because this is already getting problematic at younger age than we thought with vaccine response, right?
I'm sorry, my internet froze. Can you repeat
sorry.
that part of that question?
I just wanted to highlight that you showed already people older than 35 years old, they show reduced responses. And of course, in the much older group, you see further reduced response. But like 35 years old is not that really considered older. But
Right, yeah, that was pretty surprising finding. I mean, we definitely expected those results in the older 65 and above individuals, but to see that individuals starting at age 35, also, their responses began to wane compared to very young immune systems was pretty surprising.
Yeah, and also, so when we look at the seroconversion, we see less than 50% of people seroconvert, but maybe that's also, as you said, like not really fair, because a lot of people start very high. So maybe as vaccinologists, we should to promote vaccines better, we should have better measures to show how people respond, maybe while adjusting for their baseline titers, because indeed there's not much room for producing more antibodies because you already had protective titers. And it sometimes makes it look like these vaccines don't work that well because only less than 50% of people show seroconversion. But actually, it's really not the case, right? So that's also maybe something we should do as vaccinologists.
Yeah. I mean, that's something that's popped up in the literature the last couple of years, is that the effects of repeat vaccination might actually be a negative thing. But I think it's more of if you're getting vaccinated every year, your titers are maintained at a higher level, so there's not a lot of room for those titers to grow. Whereas if you drop back down to baseline and then you get boosted, you see a large spike. I thought that was one of the cooler findings of this paper, was that we were kind of throwing a little bit of shade on that previous findings that repeat vaccination is not as beneficial.
Yeah.
It looks like
We have one question. So the question is, how much of the age effect is due to B cell imprinting rather than aging immune system?
Yeah, so we're beginning to sort of look at that. Some other people in our lab are looking at B cell responses to the different vaccines and B cell energy, particularly in the elderly individuals. It's really hard to know what each individual was imprinted with, or their particular history to influenza. But I imagine a large part of this is due to cellular imprinting, either through vaccination or natural infection that occurs through people's lives. We have seen in other publications in animal models that we've done that if you prime mice or ferrets with viruses, let's say from the 70s, 80s, or 90s, and then give them a modern vaccine, that their responses do differ based on what they were primed with.
Yeah, I think that's a really fair point, and also quite complicated to disentangle because everyone is imprinted differently. So there are no further questions that I see on the panel. So I would like to thank you for your presentation and also for your answers. So maybe we can switch to the next talk.
Thank you.
And so next presentation is from Michael Schotsaert. He did his PhD in Ghent University, and now he's an associate professor at Icahn School of Medicine at Mount Sinai, and he's gonna talk about mentoring session. So this is the second part, is the mentoring session. So first we had the scientific talk, and his presentation is about setting up an academic lab, opportunities and challenges. So Michael, please, floor is yours.
Thank you very much, and thank you for this opportunity to present here. So, as already said, I am Michael Schotsaert. I work at the Icahn School of Medicine at Mount Sinai in New York, after I did my PhD in Ghent University in Belgium. And so I came here to New York about 11 years ago as a postdoc. And then I did my postdoctoral work, and I had the opportunity to do a lot of BSL-3 work with many viruses that were emerging, like influenza virus, later on also SARS-CoV-2 virus. I started my lab in 2020, January 2020. And as most of you know, this is exactly when SARS-CoV-2 started circulating and the COVID-19 pandemic broke out. So this coincidence of setting up a new virology lab with access to BSL-3 and BSL-3 plus facilities with a new pandemic was very challenging, because there was enough work to do, and a lot of opportunities to collaborate, but also I need to write grants from within BSL-3, let's say. So, and I relied on a very strong team that I was lucky to be able to work with from within Mount Sinai, and I will explain that in the next slides. But that's a bit to kind of illustrate how I started. So if people are setting up a lab now, that's of course different. And because there is currently no SARS-CoV-2 pandemic going on, there is a lot of flu going on, especially in Europe, I heard. So it's challenging times. And if you set up a lab in vaccinology, it's always going to be a lot of work that is to be done. So in today's talk, I just say this introduction because there is not just one way of doing this, setting up a lab. And I will share some ideas and some of my personal experience. And of course, setting up a lab will depend on the research topic you want to start in and the research environment. So for me, that was virology, immunology, and vaccinology. And most of the people here will be interested in vaccines. But of course, you may be a clinical researcher, a preclinical researcher. Maybe you're more interested in the engineering part of vaccines, like vaccine production, purification, scale-up, etc. It will also depend on the size of the department that you're going to join. A small group of, let's say, 5 people, maybe a large group for a small department, or a small group for a large department. And the size of the department will also define if you can rely a lot on the expertise of colleagues. For example, if you're the only vaccinologist in a microbiology department, then you're the expert in vaccines, but you have nobody to immediately ask a question about vaccines in your own department. If you have a lot of colleagues working on vaccines in your department, you will be one of the vaccinologists, and well, you will have some competition, but also opportunities for collaboration within the department. And of course, also, if you can rely on certain research facilities in your department or in your institute, that will also define what you have to buy. And this will also be important when you set up a lab, of course. So why would somebody start with setting up an academic lab? Many of us, and that was the same for me, at some point in your career, you may have like the question, should I stay in academia or should I go to industry? And so that's a question that many of us ask ourselves when you're in research. But of course, there are also other options. And I actually, personally, I didn't even know about these other options. You can go into policy, public health. So, but there is many questions that come up. And many of these questions, or the idea of setting up an academic lab, may be driven by a certain research idea that you still have and you really want to pursue that. Or if you're fascinated by international mobility like I was, I really wanted to leave Belgium and do something else with my science. So that was also driving motivation to go to the US. But on the other hand, setting up an academic lab may also be influenced by other choices you did. For example, you get at a stage in your career or in your life that you make other choices. You have invested or committed, for example, to family duties, or maybe you start a family. So these things are all important when you consider starting up an academic lab. And so the first piece of advice that I can give is actually that you should look for mentors and build a strong network. And actually, the beauty of ISV, the International Society for Vaccines, is that actually they can help. And actually, this is a screenshot from the website. And if you go to the website at isv-online.org, you can see under the careers tab, there is a sub-tab, mentorship, and there you can find some guidance. And the ISV is actually providing a form where you can say what you want to do in academia, in industry, or if you want to transition from academia to industry. So there we can find mentors, or ISV can find mentors to help you with that. And so with that, we get more into detail on how to set up an academic lab. And so I will discuss a couple of points. So the first one is scientific management. So what do you want to do when you set up an academic lab? The second one will be the lab management. So what do you need to do that? How do you do that? So what is the kind of environment you have to create to answer the scientific question that you want to answer? Very important is, of course, the budget. You need money to do all of this. And so you have to know what will this cost and how to manage the funding. Personnel management, which is maybe the biggest of the most important parts. So who will you need to do that? You cannot do this on your own. You cannot be a one-person academic lab. So how will you motivate people to work with you on your ideas? Yeah, and how will you make these people work with each other to achieve the goals of the lab? And then very important, as you see, there is different points you have to address. So time management will become extremely important. And then finally, I will say some things about visibility and networking. So for the scientific management, so what you typically start with, of course, is you define the research goals and the objectives. What are you going to study? What will be the primary focus of your research? And ideally, you find your niche, because if you're going to do the same as what other academic labs will do, then you're immediately going to have like competition, and it's going to be very hard to stand out with your academic research, especially because you're starting the lab, and established laboratories have this advantage of already 10 years or more of experience. So your niche can be a specific disease. There is many emerging diseases for which we don't have vaccines yet, or no good vaccines, so there is definitely a need for that. Maybe you have a good platform for vaccines that you want to kind of develop further, or you want to kind of test how you can bring this to the clinic, for example. Or maybe you're focused on a very specific product. Many laboratories have been focusing, for example, on universal vaccines for flu. Afterwards, it became for SARS-CoV-2, etc. So you can have a very specific product in mind as well. But overall, you have to break out of your scientific comfort zone. If you set up an academic lab, you start something new, you can also kind of suggest new things that have not been explored somewhere else. So basically what you can do is you can define high risk, versus low-risk projects, and make sure you have both of them. And the high-risk projects may be the most fun, but also they're risky. So if they fail, it may be that you end up with no data, no data that can be published, or no product. So therefore, it's good to actually also have low-risk projects. And I will go into detail what those are. But low-risk projects are typically almost fail-proof and actually give you data for maybe not the highest impact journals, but maybe they're very good also for collaborations to answer questions that industry has for collaboration with you, etc. And also something that I want to stress is invest time and money in data storage, management, and electronic lab notebooks, because you may produce the most amazing data. If it's not stored and if it's not published, then actually you're wasting your money. So how can you now define the expected outcome, scientific outputs for your lab in short and long term? And that's something you really have to spend time on. And so a typical scientific output for an academic lab is, people think of papers, but you can also have, of course, patents, and patents can be as important as papers. I mean, they're kind of a form of publication, but also they protect your research, and that might be important for both for yourself and for your lab, so that somebody else cannot just or industry cannot should recognize you when you kind of publish this. But also it gives you kind of a nice position to collaborate again with, for example, industry, so you can license that product, and then this can also allow you to do more research in the lab. Another very important scientific output concern is how many students can graduate from your lab, how many postdocs do you want to continue a career in academia after they were in your lab. Collaborations with industry can be very interesting and also be a very good source of funding for your lab. How many products do you want to see in the clinic eventually? That can be a goal of the lab. Maybe you want to start a lab with the idea of creating a spin-off company later on. And maybe you want to be very successful in funding acquisition, so you can do whatever you want, let's say, in the lab. So all of these kind of points are actually measurements of success for your lab, either in the short or in the long term. And that's also important to kind of stress all these points if you want to, for example, make promotion within your institute. So it's very important when you set up a lab to kind of start thinking about these things early on. Budget management, of course, starts with getting the money for doing this research. And so you will need to consider several funding sources. In academia, research grants from funding agencies are a classic to go to, but there's also a lot of philanthropy that can actually fund part of the research. Sponsored research agreements through industry collaboration, as I mentioned before. There might be institutional grants that are not from funding agencies. It's very specific for the institute, but it's definitely worth looking for them. And when you work with postdocs and students, very often they can apply for fellowships that can cover their salary and some of the bench fee. You can also ask lab members to apply for travel grants, awards, because sometimes this also comes with money for the lab. And of course, if you start a new lab in an institute, you may be in a position that you can negotiate a seed fund, which is an unrestricted fund you can use for starting up the lab. That's very important also. So for budget management, it's important to understand the numbers. So one of the biggest costs will be personnel costs. You have to understand this is salary, which consists also of fringe benefits and costs associated with, for example, visa application if you work with international personnel. And you should also consider long-term retention strategies to avoid a costly turnover. It typically costs more to replace somebody than to be able to keep somebody. For example, if somebody comes to your lab with a fellowship and the fellowship ends, it's a good idea to be able to kind of continue the salary, of course, so that this person doesn't have to go back in the middle of a project because the fellowship ends. Equipment is of course also a large cost, so you should identify the major equipment that you need to, and maybe you can actually do some cost saving by buying refurbished equipment, like by using shared facilities, by lease instead of buy a machine. Another big cost, which is more volatile and can change very fast, are supplies and consumables. This is budget that you need to kind of have for recurring costs, like for example, pipette tips, reagents, glassware, things that actually are used all the time. They're necessary, but yeah, you will have to buy them all over and over again. So ideally, instead of having a very detailed budget, what I try to do is to have an estimate of the monthly expenses based on the planned experiments. So I have like a magic number that basically allows me to quickly see how we are doing in the lab. So because if you have all these detailed budgets, it becomes very difficult to manage and to monitor. But if you have magic numbers, if you know more or less a postdoc will cost this, and a student costs this, and research associates will typically be this, and then the equipment will be this, we need to budget a little bit for maintenance, etc. Like this, you can get a rough idea of how much you have to get get us from a research grant or something to kind of budget correctly for the next year. And also it's important to categorize needs into essential and non-essential, because this will allow you to prioritize the spending. Often forgotten costs are actually that you have to buy laboratory furniture and storage solutions. It's crazy, and I didn't realize it in the beginning. It's crazy how much storage solution you have to kind of, or how much you have to store. You have to buy all these products and kind of put them somewhere, right? Safety equipment and protocols take up a lot of money and are typically forgotten in the beginning. Maintenance and operational costs. You can buy a very expensive machine, but you will also need a maintenance contract to kind of keep it running. And facility user fees also take up a large part of the budget if you rely on facilities, of course. As I mentioned already, you can explore cost-saving measures, for example, shared facilities, but also if you set up a new lab, very often you can benefit from discounts from vendors because they do this for these special actions for new labs. Also build some contingency funds because you will encounter unexpected costs. A machine breaks, you need to hire somebody because you need more hands for a certain project, an accident happens, etc., etc. So build some contingency funds. And also very important, try to track the spending and adjust if possible. If you know what you're spending and if you know where the lab is bleeding money, that's where you have to kind of intervene, of course. If you don't monitor, if you don't keep track, it's very hard to find this. Or you find it out, but it's too late. Very brief, I can say something about laboratory management. Of course, when you're going to start a new lab, you will be able to kind of organize it the way you think will be best. And so you have to start thinking about space planning. So ideally, you can assign one bench and a desk per researcher if you're a wet lab. It is very important to respect the kind of environment for researchers. Everybody has their own kind of needs in terms of privacy, silence of working. Like everybody has their own way of working on a bench. So it's something that takes time and is worth spending some time for, because otherwise you may have to kind of correct it afterwards, and then it's harder because you will have already organized your lab with certain machines in certain space, and then it's harder to kind of fix. So ideally you can include some common spaces for shared equipment. You can optimize the layout for a workflow efficiency with research stations for specific tasks. For example, if you do a lot of preclinical work, you may not want to have a biosafety cabinet where a lot of necropsies are done for animals next to the desks where people are working on their computers. Liquid nitrogen tanks that are in the way, freezers that are making a lot of noise next to a desk. That's something you have to keep in mind. And yeah, so ideally the workflow allows that the wet lab is somehow separated from the desk space. On the other hand, if the desks are too far away from the wet lab, that's also kind of a problem, because then people will lose time going from the bench to the desk, etc. So these are things you have to think out before you organize the lab. On the other hand, lab organization is not always that easy if you work in existing labs that are already kind of designed for certain with a certain layout. For example, in my case, I work in New York in an old tower, so we don't have much space to kind of rearrange or to move facilities, etc. That's not easy. Again, the storage is very often a problem or something that you have to keep in mind at least. And storage takes space, so you have to kind of look in the design and the layout where you're going to store all that stuff. And then placement of safety equipment like fume hoods, eyewash stations, and fire extinguishers is of course extremely important. And the fire safety, for example, will also come into the lab and actually tell you where you need more of the fire extinguishers or eyewash stations. So also keep in mind that you will have to kind of have space for that. Then there is some other safety measures that you have to keep in mind when setting up a lab. For example, you will have to develop a chemical hygiene plan. You will have to have and stock personal protective equipment, PPE, like lab coats, gloves, goggles. You will have to train personnel on safety protocols, which is it's very important, of course, but also takes quite some time and a lot of administration as well. For example, for handling the hazardous materials, that will require, for example, special cabinets. Again, it's an extra cost, and it requires extra organization for the layout. You will have to set up emergency response procedures and train the people for these. So all of these things take a lot of time and administration. Waste management, it may not always be that it may not always seem that important, but actually, especially for a virology lab or a microbiology lab, waste management is of course very important and will take up a lot of money and time from your people. So then for the personnel management, this is maybe the most important when you set up a lab after having the funding. So you will work with different people, and they all come with different expectations and with different goals. Like a PhD student wants to do the PhD and do the science. A postdoc may want to continue on a project that he or she started before, or they started before, and then maybe they have like a very specific project in mind. Whereas research associates might enjoy the science and do the work, but also they see this more long-term as like a long-term job that they want to do for maybe even 10 years or longer. Whereas PhD students and postdocs may see this shorter in time, especially PhD students, of course. Some people like to collaborate a lot, and they are very fine and do very well in a large group where people divide the tasks, but some people prefer to work and do many things themselves. And both of them are okay, but you just have to keep that in mind and try to organize the science so that this is possible. I have always seen that it's best that you can encourage collaboration within the lab, keeping in mind that some people prefer to do certain things on their own, but still collaborate for other things. But if you consider collaborations, you should consider that these are also training opportunities. And so you can team up a senior lab member with new recruits, for example. If you encourage collaboration, you have to keep in mind that this may result in shared authorships on papers. But of course, you will develop a lab that has a lot of shared expertise. That's also nice. And that's why I also encourage to recruit people with different backgrounds, training, and interests. For example, in our lab, we have veterinarians, we have biotechnologists. I'm an engineer by training. We now have a medical doctor. So these different backgrounds, they give feedback to each other and allow to train each other on new techniques or give new insights. It's very cool. Consider training opportunities over productivity from time to time for your lab members, because yeah, you're an academic lab, of course. Training is important, and we're not working for a product typically. We try to train people and bring them to the next level in their career. And to help you out with all of these things, it's very important to hire a skilled and motivated lab manager, or even more than one lab manager, for helping you with setting up the lab and actually managing all these components of what I just mentioned. And also I want to say that there is always an optimal composition and size for every lab. It's something that you will find out as you go. If the lab grows too fast or too large, then it becomes hard to kind of manage everything. If it's too small, you don't get the things done that you would have to do for the funding that you promised. So there's always an optimal composition. And I think in the beginning, it's hard to see or to define how that is. You will grow, the lab will grow organically, and then yeah, you will find out. Then very important is time management, because I mentioned all these things. So there is very little time for the actual science once you start up a lab. That's very important to understand. You will be applying for funding. You will do a lot of administration, mentoring and teaching, training people. It's very important, writing manuscripts. So basically, it's very important to manage your time as well. And important also in that perspective, because people come with different expectations to your lab. You should assign projects that can be finished within the duration of the research day. So if people come for 1 year, I mean, the project ideally is also finished or doable in 1 year. If people are there long term, this can be more long-term projects. Or people should have parts of a project that can be finished within the time frame of their stay. And then finally, collaboration, networking, and visibility. It's very important that people know the lab and that they know what you're doing. So you can engage with other labs or institutions to share resources and expertise. You can build an online presence with a lab website or social media announcements to attract collaborators and students. presentations at local international meetings, and go to networking events. It's ideal to get to engage with industry partners, for example, that may be looking for specific expertise. It may result in a research agreement. You may find new people. You may find the next job for your students, etc., at networking events. And so with this, I want to conclude. And as I mentioned, there are many aspects that you need to consider when setting up your laboratory. It's not always scientific, but the satisfaction from seeing people thrive in their careers and being able to be part of that journey is very fulfilling for me personally. And so with this, I want to thank you for your attention. So my lab is now 12 people, as you can see. We are present on social media and the internet, of course. And we can actually, I invite you to come and join us in Stellenbosch in South Africa for our annual meeting, where we can do some networking and discuss all of this in person. And thank you very much.
For this great presentation, Michael, is you really put it down point by point for each management section. I think it was very useful for a lot of people. So we have some questions. So maybe I'd like to start with the first one, then I will also have a question for you. So one question is asking, what is the thing / person that helped you the most developing your management skills? Do you have any practical suggestion?
So that was one of the hardest parts for me, because as I mentioned, the lab started during the COVID-19, so I was mainly in BSL3, and I was not able to talk to mentors or look for mentors. But actually, what I was doing then, I was reaching out to people with specific questions. I was looking for my mentors, and that is also what I mentioned in one of the first slides. It's important to find a mentor or multiple, and just with specific questions you can go to. For me, that was older professors here in Mount Sinai, but also people that I was talking to, just like, I mean, to be honest, my wife, she's a therapist, and also she was working at the university in Antwerp, and so for personnel management, she was actually very able to give me some good feedback, because all of these aspects, when you start a lab, is actually they require a lot of different angles. Like personnel management is something that, I mean, you can also ask other people outside of science. It's more because basically you're setting up a small business. So in a small business you would have the same problems, and you would also need an HR department, let's say. So these questions you would address to, or you would ask to other people, and then the scientific questions I would go to my old mentor, or I would go to other people that are that are known in the field for this. So basically look for mentors. Again, ISV can help with that, with specific questions, and then yeah, try to do the best, right?
It sounds really challenging, especially like during Corona times. I did my PhD during Corona, and it was quite crazy, and I cannot imagine how you went through all these things, starting a whole new group. So it's really impressive, and it's inspiring, because now you are really there, did that, and telling us people how it all went. So there's another question, is as a postdoc, when is the best time to start learning about these management skills? Because it's like starting a company indeed, but as a postdoc, most people are very busy with output, doing research, publishing papers, getting grants. But this is sort of like the unseen part of the starting your own group. And I can imagine this is not something that's one person sort of learn in I don't know, in a month or so. So when is a good time to start getting into this, or how would you advise postdocs who are thinking about starting this journey, where to start?
Yeah, so from my personal experience, I saw that the task of a postdoc is very different depending on the institute. So I would say as early as possible. The moment you start working or have the chance to train people and work with people, and typically as a postdoc, you will have, for example, a grad student that you can mentor. I mean, the moment you start working with people, you kind of get these people skills. I mean, it comes in handy all the time, of course. So I would say as early as possible. I know that some institutes actually have nice courses to kind of also train you in some of these things, like people management, but also budget management, IP. So try to do as many courses as soon as possible. I know that, for example, when I came to the US, the tasks of a postdoc were very different. So all of a sudden I had to do everything individual. So I was not working anymore with students too much or with other postdocs. I was doing more things individually. And then, yeah, you don't get a chance to kind of develop these skills too much. But then as I was moving on in the career and I started working again with people, yeah, then this came in handy that I did that before, for example, in Belgium or in other places. So I would say as soon as possible. But also a postdoc, I think, ideally should have the chance to kind of develop their own science. So they can actually take that further into the lab that they want to start. So if your task as a postdoc is only mentoring people and not doing science, that is also maybe a problem. So I think you should find a nice balance between your own research and mentoring people and developing these skills. It will also depend a bit what is the plan of the lab or where you want to take it, right?
Yeah, thank you. I think it's really important what you mentioned, because people can sort of get carried away with either mentoring a lot or doing a lot of science, but these things can be, I think, forgotten. But it's also a big chunk of work for the next steps. So there's another question which is not completely clear to me, but I will read it. So the question is from an anonymous person saying that, is it a good idea to start a lab with your savings before getting grants? And I believe I'm not 100% sure what they mean by savings, but this is the question.
So if the savings mean personal money, I would personally not do that. It is a bit the same question as, should you set up a company with your personal money? I think you should consider that. If you would answer yes to that question, I would personally say no to that question. I think, again, if you want to set up an academic lab and you have a certain technology that you want to take somewhere, I think it's a risk you take, but it will depend if you can. I mean, I would personally never put my personal savings into a lab because it's too riskful. Because it's still science. It's possible that the lab may not be that successful after a couple of years despite hard work, for whatever reason that is beyond your control. So I would personally not advise to use personal savings. Of course, that is a personal thing then. Yeah.
Yeah, and I think the important thing is we are talking about quite large amounts of money to start a lab.
Yeah. Yeah, if you want to have an idea of a budget, the personnel cost for 10 people in the US, I can tell you overall, is between $1 million and $1.5 million for 1 year.
And this is just a personnel, because you need the machines, you need everything. So it's maybe that's gonna make it clear for the person who asked the question. Like, we are talking about millions in this context.
That's of course for a mid-size lab. But yeah, you have to think of hundreds of thousands of dollars per year, even from the beginning with a small lab. Yeah.
All right, thank you very much for your talk. And also, again, I would like to thank James as well for his presentation and all the attendees. So it's nearly 5 o'clock, so we're gonna conclude. And make sure that you join our next webinar, 6th of March. And in the chat, you can find the link to our LinkedIn group. So you can also join us with organizing webinars, networking, and we also have this mentoring sessions and even more. So just take a look, get in touch with us, and we can give you more information. And with that, I would like to end the meeting, and I wish everyone a nice weekend.